talk by Miguel Teixeira, Biomimetic Oculomotor Control. Welcome, Miguel.
Yeah, that would be me. Uh, that was, uh, Hello, everyone. Uh, I hope you can see my slides.
Yes, yes.
Okay, perfect. Okay, thank you. Hello, everyone. Thank you for having me. My name is Miguel Teixeira, and from the next 15-ish minutes, I'll be talking a little bit about the work developed by both Professor John Van Opstal, Alexandre Bernardino, and me on the biomimetic control of um, um, a computational model to perform rapid eye movements. These rapid eye movements, also known as saccades, which are here represented in the video, are extremely important in localization tasks. You see, the reconstructed images by which we perceive our world have not the same resolution all across our visual field. This is because the light sensing cells, which effectively sample the visual information from our environment, are not homogeneously distributed across the retina. In fact, the area that we associate of having the highest degree of visual resolution, called fovea, corresponds to a really small portion of the retina. Therefore, to better understand any particular region of the environment, we must reorient the fovea so that it aligns with that region of interest. Saccadic motion, therefore, function as a very convenient way of performing this job. This is because these saccades are fast but also really accurate eye movements, which means that the eye can reorient really quickly to any particular region of the environment that we want to perceive. Saccades have been a hot topic of research over the past years, and now we know that they are quite stereotyped movements, meaning that they follow certain rules which are equal for even for different individuals of the same species. Nevertheless, the underlying mechanisms by which these motions are created are still not well understood. In this work, therefore, we set out to replicate saccadic-like movements, which I will briefly explain, using a biomimetic eye model, here represented in this picture. Note that this is a 6 degree of freedom model. We believe that by studying how saccadic motions are generated on this virtual environment, we can then use this information to better understand how saccades indeed emerge in the real biological system, and later use this knowledge to create better robotic designs. Sorry. In this work, we drew inspiration in many different ways from the real biological systems, and therefore some important informations should be explained. For instance, the eye itself is constrained in moving in rotations, which are driven by 6 extraocular muscles, here represented in these elements here that I'm pointing, that attach to both the eyeball and the skull in a non-symmetric configuration. Moreover, the interaction between this organ and the fatty tissue that encircles it makes this an overdamped system. Note, although that despite of this overdamped nature, saccadic movements are known to occur over time scales much shorter compared to the characteristic time constant of the system. This behavior is actually induced by a particular activation signal that is applied to the brain, applied to the muscles by the brain, which has a particular shape. It has this pulse step shape here, which allows the muscles in the system to both overcome friction and elastic restoring forces. An interesting point to note regarding this pulse step is that it actually originates from a single pulse, which then gets integrated and summed to a scaled version of itself, here depicted in this block diagram, which is known as Robinson's model. Well, in the beginning of this presentation, I told you that the objective of this work is to reproduce saccadic-like movements. And what we mean by this is that we want these movements to have these 4 particular characteristics, which we know the saccades follow, which are the fact that saccades actually follow straight trajectories, meaning that the eye rotates about a fixed axis of rotation. We also want to, for the main sequence, which is a set of relationships here depicted in the slide, to emerge. Also, it is interesting to note that despite of the muscles in this system being mechanically independent from each other, and therefore theoretically they could be uh, they could be controlled each separately. The controlling strategies that are applied by the brain naturally groups these elements in pairs, with the elements within each pair acting in an antagonist-agonist manner. This is that when one muscle contracts, the other will tend to relax. Lastly, it is also interesting to note that even though only two degrees of freedom are required to specify any given gaze direction, the azimuth and elevation, the rotation of the eye about this gaze axis, the cyclotorsion, is actually constrained as it was discovered by Donders. And Listings later refined this observation by noting that this orientation of the eye actually is constrained to a plane. Now, we want in this work, again, to create movements with these types of properties without directly imposing the eye trajectories. And so this begs the question on how can we generate, how can you create this generation process? Well, we started by noting that saccades are hypothesized to derive from a speed accuracy trade-off, and therefore we model this saccadic generation process as an optimization procedure. Following our methodology, we have an agent which is able to act on an environment with unknown dynamics solely based on information regarding the initial eye orientation and the desired eye orientation. The environment itself will only output a reward signal, which is a scalar quantity that incentivizes the agent to create actions that result in higher and higher rewards. The optimization process, therefore, consists of a predetermined number of interactions between agent and environment, which we individually denote as episodes. Briefly, in each episode, the agent is able to choose a total of 24 different parameters, which are then decoded in this pulse step generator. This element functions similarly as the Robinson's model that I previously described, and therefore a pulse is first created, it is then integrated and summed to a scaled version of itself to create this pulse step. Note that each individual pulse requires 4 parameters to be fully determined, and since we have 6 independent muscles, we have to have 6 of these elements. Also note that 6 times 4 equals 24, which explains the figure that I previously mentioned. The output from these elements will then act on the simulator, which is able to estimate an eye trajectory, which is subsequently processed in order to compute the aforementioned reward signal. This reward function in this work consists on weighted sum of metrics such as the accuracy of the motion, the duration, the energy spent during this motion, and the tension applied by the muscles once the eye reached its final orientation. In the beginning of optimization process, the agents would typically perform random actions, and therefore when tasked with reaching certain targets, here depicted as the square markers, it would not be able to comply. Nevertheless, once the optimization procedures start taking place, we can see that the rewards overall would increase over time, and we can see this by noting that the agent can now reach the targets. By the end, the trajectories were quite straight, which is quite interesting because this was never directly imposed. Again, I restate that straight trajectories in a way means that the eye rotates about a fixed axis of rotation, which in and of itself hides an interesting characteristic regarding the horizontal and vertical components of the velocity profile, which is that they must cross-couple. Theoretically, this cross-coupling would follow this cosine dependence, here depicted as the black dashed line. And if we compare this theoretical this theoretical model with the data that we obtained from our simulation, here depicted as the colored solid lines, we can see that they overall match quite nicely. Turning our attention now to the main sequence. Here I plot these relations using data obtained real experiments performed in humans in this case. And comparing this with the data that we obtained from our simulator, we can see that they overall also match quite nicely, and the expected behaviors are indeed followed, with the duration of the motions increasing with the amplitude, the peak velocity saturating over large saccadic amplitudes, and the product between peak velocity and duration following this linear dependence with the saccadic amplitude. Again, comparing these two relationships, which were plotted using different data, we can see that they overall match quite nicely, which is quite interesting because, again, I restate that this was never directly imposed in our optimization procedure, and it naturally emerged. During when we developed our computational model, we took great care into making it as biologically accurate as possible, and therefore we created it such that the muscles could be controlled independently. Nevertheless, the control strategies that emerged from this optimization would naturally group the muscles into pairs with the activation signals applied to each muscle following an antagonist-agonist behavior. As we can see here, when one muscle contracts, the other will tend to relax. And this is what we also observed in the real biological system. We further tested this hypothesis and noticed that the correlation between the activation signals applied to these muscles were computed to be really close to -1, which would imply that these activation signals actually oppose each other, as it would be expected by the antagonistic behavior. Another interesting point that we discovered was that different type of muscle pairs would typically be employed to perform different movements, with horizontal movements being mainly created by one type of muscle pair, whereas purely vertical motions would require two. Lastly, we also wanted to confirm whether Listing's law was valid in our simulator. Again, I restate Listing's law states that the torsional component of the eye, the horizontal component, and the vertical components should all be clustered around the plane. Here I plot this information. This data was obtained using, in this case, monkey subjects. And here I plot two views of this plane. Here we can see the positions of the vertical and horizontal eye positions, and this tells us that the monkey itself created saccades all across its oculomotor field. Note, however, that despite this being from -22 to 60 degrees, the torsional component of the eye remained pretty closely scattered around 0 degrees of the torsional component. Again, comparing this with our simulation data, we can see that there is a high degree of, they are quite similar. Again, in this case, our agent was tasked with creating saccades all across its oculomotor field. You can see it -40 to 40 degrees, horizontal and vertical. However, the torsional components remain pretty closely around 0, which is again really close to what we would expect a living organism to perform. So in this presentation, in this work, I showed how we developed a computational model of a human eye using 6 degrees of freedom and controlled it with biological inspired control signals. We also empirically validated a model-free methodology to control these kind of systems with saccadic-like movements emerging from this optimization procedure without directly imposing the trajectories that we know are correct. Thank you very much for your attention, and now I'll be open for questions if you have any. Hey.
Hello. Yeah. Thank you
Yeah,
very
hello,
much.
I'm here.
Thank you very much, Miguel. Um,
No,
yeah, now it's a question time. So one question I was wondering whether are you using any eye tracking mechanism for learning the saccades, the movement?
no. The only mechanism that we are using for learning, so this is purely computational. So this is we have only this model, which I showed here in this slide. Yeah, there we go. This is purely computational. This is a visual representation of our computational model. And what we are doing to learn these saccades is that we are um, asking our agent to reach a certain goal, and um, we are giving the agent a reward which is based on how optimally he reached the goal.
So this
And
is
we are
RL.
using a reinforcement learning methodology. Yes, yes.
Yeah, I was asking whether it's RL you are using. Reinforcement learning.
Sorry.
No, I was asking
Yes, it's
whether
reinforcement learning. Yes, yes.
yeah, okay. So is this any particular simulator are you using? Is
Yes,
this
this
any
is a
particular
simulator based.
simulator? What's the name?
Sorry, could you repeat? My audio is quite poor. Sorry. Oh, the simulator itself, it was we that developed from the ground, so it's not it's not it's not publicly available. It was we that developed the simulator itself.
Oh, you developed from scratch.
Yes, 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 yes.
Oh,
It
nice.
was not me. This is an ongoing project. This is an
Going
ongoing
project.
project, and I I was the last iteration of this model.
Oh, okay, okay. And yeah, at some point you were mentioning that you will be comparing against the human saccades, right? In the
Yes,
biological
yes.
vision. So how will you collect such data? Um,
Yes, so so one of my supervisors is a neurophysiology, and he was the one that collected this data. For instance, here, uh, these data here were collected by him, and I just compared it with the simulations that I perform. Yes.
okay. Any other question from the audience? Audience question? Okay, I think that's all for the questions. Thank you very much, Miguel.
Thank you.
Thank you
Thank
for
you for
coming.
having me.
Yeah, yeah. And next we have Gonzalo Mesquita.